Well, for those who don't know me, my name is Fernando. I'm a collaborator here for the USDA IRS, working at Harbour Branch. And again, I want to thanks on behalf of Harbour Branch and the USDA for your participation in this meeting. And this presentation, we will basically share with you the results of the last few years. And just as you know, we will not go into detail, but rather focus on the results of the experiments. And in any case, this presentation will be available for any of you who is willing to have it. So I want to start acknowledging that all this work is the result of a collaborative effort between the USDA, IRS project objectives, the farmers' priorities, and the collaborators. Among those collaborators, we got the Universities Research Institute as Hapsi World, Bosman Fish Center, MODE, University of Maine, University of Barcelona, and industry as Aquatic, Stradium, Aqua, Live Atlantic Space. So I'm going to start for the USDA. There are three main objectives for these projects. The objective one was develop expanding strategies and agriculture methods. The objective two is to increase the understanding of fish physiology of fish to improve management strategies. And the objective three is to develop methods for genetic improvement. Regarding the farmers' priorities, their concerns rely on regulation, diet, disease, new species, and density and temperature. So with respect to the regulation, we and the industry already acknowledged that this was not the project for that change. So farmers' concerns on diets were stated in three topics, mainly on feed manufacturing ability as the lack of commercial diets, cost-effective feeds, or a second topic regarding diet development as developing specific diets for red drum or pompano, and a third topic as applications and feed technologies regarding frequency of feeding, research on farms, or minor changes to existing diets. So regarding the topic of disease, there are two main aspects that farmers were concerned, and is diagnostics and treatments, and disease types and species susceptibility. Last, we have the farmers also expressed their concerns about potential future farm species and the relationship between density and temperature. Now, Alison will proceed with the first part of the results regarding broodstock and agriculture.
Hi everyone, I'm Allison. I'm a researcher here at Harbour Branch, and today I'll be talking about some of the broodstock diet projects that we have done to accomplish our objectives. Broodstock diet research is especially relevant in aquaculture because it is a huge factor in areas like reproductive success and can greatly impact things like egg quality and larvae quality. So I'll start with the study we did with Florida Pompano. So from 2022 to 2023, we conducted a broodstock diet experiment to find an alternative to cut bait. We tested a commercial diet from Breedem and a pelleted diet that we make here with an added 10% IMTA. In terms of fatty acids, the alpha diet eggs had higher N3 PUFAs and higher DHA to EPA ratio. These are all known indicators of good egg quality. So in conclusion, the alpha feed was able to achieve same or better quality of eggs compared to the control and Breedem. Alpha as a feed ingredient has the potential to improve spawn quality.
Thanks, Rebecca.
So this research project was part of our student Flavia Perron's master's thesis. She graduated last December. This work was also presented last year at Aquaculture America. So we also worked with Hub's SeaWorld, where we did a broodstock diet study with California yellowtail, which I will be referring to as CYT, to replace cut bait with a commercial diet. We tested two commercial diets previously used by CYT producers and researchers, by Oregon and Breedem. So here are results. The control diet had the most spawns, but it had poor spawn viability and hatch rate. For the Breedem diet, it produced eggs with large egg and oil diameters, but it had very few spawns and abnormally low hatch rates. The BioOregon diet had comparable spawn quality to the control, but it was only tested on one tank, so there are insufficient replicates to confidently make a recommendation. So in conclusion, Breed M is not suitable for a total replacement, but it can be a good supplement for ARA and EPA. BioOregon was only tried with one tank, but we have very promising results, so it should be further tested. We also found that all these diets did not meet qualitative requirements for DHA, which could have affected the spawn quality in these studies. So this project was completed and presented at the Women of the Water conference in 2022 at MOAT, and it was also published in the North American Journal of Aquaculture as a featured article last year. So from that study, we wanted to zoom out and focus on the bigger picture. So how long exactly do we have to feed a diet to the fish to see it being reflected in their eggs? So this information can be very useful for when farmers want to test out a new feed, but they are not really exactly sure how long it will take into effect. So we did a diet shift study with CYT to answer this question, where we fed an alternating diet of Vitalis and cutbait through the spawning season, and we compared spawns from each diet switch period. So we found that it does not take very long at all for eggs to start incorporating a diet. And you can see that here in this fatty acid scatter plots, where the trend drastically switches to the opposite direction shortly after the new diet is introduced. Our results found that producers should wait a minimum of 10 to 19 days to allow for nutrients in the diet to be reflected in the eggs. We also found that LNA, LA, and EPA, which are very important omega-3 and 6 fatty acids, can be stored by the fish to account for future fatty acid deficiencies. This was also the first time we tested Vitalis on CYT, and we had very comparable spawn quality to cutbait. So this work was part of my master's thesis and was presented in several aquaculture conferences, and we also published it in Aquaculture this year. So on to our larviculture projects. So one of the largest bottlenecks in larviculture is the use of live feeds like rotifers and artemia. Farmers are very dependent on them to bring larvae to weaning, and they are also very prone to crashes. An ideal alternative would be to use commercial diets instead and eliminate the use of live feeds, which is the aim of the study. We use the Inve Natura diet here. This diet was previously used to replace live feeds by over 50% in sea bream, but this is the first time that it was tested in the Florida Pompano. So last year, we tested this commercial diet at Pro Aquatics and LAB at several replacement percentages, along with a control diet. We got very promising results that showed partial replacement of rotifers and artemia was possible with the Inve diet. But survival rates did drastically decrease in our treatment with the highest live feed replacement. However, at the end of the experiment, the weight and lengths were similar in all of the replacement treatments, with the control doing just slightly better. So this year, we actually repeated the experiment at Harbor Branch, but using a much higher replacement percentage. And this is to see if a complete live feeds replacement was possible with the Inve diet. So our results was that tanks with total replacement experienced total mortality at 19 and 20 days post-hatch. Larvae in our 80% replacement treatment was successfully weaned at 20 days post-hatch, just 5 days after our control. Survival rates at the end of the experiment was also half of the control. So this shows that the Inve diet is not suitable for total replacement, but it can definitely be used at a very high partial replacement at the expense of slower growth and lower survival. The Inve diet cannot replace live feeds, but it can be useful in situations when producers don't really have reliable or consistent access to live feeds, especially during emergency crashes. So this is a picture of our team on the final sampling day. Our on-farm results were presented this year at the San Antonio Aquaculture Conference, and we also had a lot of interest from the audience. We plan to do a similar study, but using red drum larvae instead. Thanks for your time and attention. Fernando will now be presenting the next part of the presentation.
Oh, hi again. In this part, I'm going to focus on the results addressing those priorities regarding the diet development as specific formulations or nutrient requirements for pompano or red drum, with feeding trials using clam meal, black soldier plant meal, different carbohydrate sources, methionine, and also priorities regarding fish management with the culture model for Florida pompano. So overall, the objective of this part is to formulate specific diets to improve growth performance and to reduce feed-related costs without affecting the nutritional balance of the diet. And among the ingredients that we tested, we have a protein source as clam meal byproducts, or alternative protein source as a black solid fly meal, or different carbohydrate sources as a whole wheat flour, wheat starch, or methionine supplementations. So regarding the use of clam meal byproducts in diets of Florida pompano, our results show that fish fed 10% and 20% clam meal replacing fish meal diets, the column green and orange, presented actually higher final weight than the control diet. So taking a look into the growth performance parameters, we can see that the fish fed 10 and 20% clam meal show the highest final weight and weight gain compared to the control diet. And also fish fed 10% presented a higher protein efficiency ratio. Overall, these results indicate that the right percentage of clam meal byproducts supports growth and benefits Florida pompano, advancing also in the formulation of specific diets that enhance the growth, suggestibility, and also possibly reducing spread of waste. In the same line, the objective of this new experiment is to determine the adequate carbohydrate source for Florida pompano. Among the tested diets, we have whole wheat flour, wheat starch, whole flour, corn starch, or dextrinite corn starch. The results show that among the tested diets, the whole wheat flour is the more adequate carbohydrate source to include when formulating diets for Florida pompano, with a higher weight gain or higher percentage weight gain. Overall, again, choosing an adequate carbohydrate source for pompano is important, as it will determine the digestibility level, the fish growth performance, and the waste load, or also the water parameters, or even the feces properties. Here, another experiment to determine the effects of replacing fish meal with different levels of black soldier fly meal. Black soldier fly meal replacing fish meal at 0% will be the control, replacing fish meal at 25%, 50%, 75%, or 100%. The results show that a fish fed a control diet and black soldier fly 25 and black soldier fly 50% presented similar final weight. So again, taking a look into the growth parameter performance, we can see that optimal parameters appear in control, and those fish fed black soldier fly meal also with a higher protein efficiency ratio in fish fed black soldier fly meal, which corresponds to the similar values in feed intake in control, 50 and 25%. Overall, what this end of these projects are to determine that choosing an adequate protein source and inclusion level is important, because it also will determine fish growth, and also will potentially reduce the waste loss, and it also improve the testability. So again, it will be a step forward toward formulating specific diets for red drums. In this slide, I want to call your attention, because another factor when working with assessment of new ingredients to include into the diets, whichever diet for whatever species, is its palatability, meaning how much does this new ingredient the fish likes. So here we performed a palatability experiment with inclusion of black soldier fly meals at 25%, 50%, and 100%. And we can see that fish fed the black soldier fly 50%, presented a different palatability response with respect to the control. Again, this shows that replacing fish meal at 25% does not have a negative control as in the palatability response of the diet. I want to also to call this attention because this is serving as simple as could be in this slide. It's implies a lot of men work, a lot of effort, a lot of time producing the feed, and actually took us 6 months more. So this feeding trial is still in process of data analysis, and the objective is to evaluate different forms of dietary methionine supplementation. But why methionine? It's because normally commercial diets are formulated with a plant-based ingredients, and they are low in methionine. So supplementing that methionine will overall create optimal diet for pompano. Lastly, we released a manual for culturing Florida pompano, and the intent of this manual is to provide fish cultures with information necessary to produce Florida pompano from breeding to harvest size, and the manual includes larval culture development, live feed culture, brood stock management, system designs, and also market analysis. So now, I will take over this part.
Hey everyone, I'm Carlie. So I'm gonna continue with objective parts B, 1B and 1C. So this first project, we were looking at supplementing diet for Florida pompano with symbionts, or I'm sorry, symbiotics, which is the combination of prebiotics and probiotics. The difference between those two things being that the probiotics are the actual live bacteria that live in the gut, and they help improve digestion and immunity. And the prebiotics are what feeds them basically and keeps them going. And those nutrients happen to be carbohydrates, and the probiotics also help break those down. So we used a species of Pediococcus, hope I'm pronouncing that correctly. And here's just an overall view of our microbiome results at the end of the experiment. So we used different combinations of, or different concentrations of symbiotics, and long story short, there was really no significant difference made by the symbiotics. We just happened to observe some increases in 3 types of bacteria, and those were in the samples that had fully supplemented diets with symbiotics. So it would take some further research, like everything else, to tease that out some more. Moving on, we investigated the effect of copper treatment on the microbiome of the aquaculture environment for Florida pompano. This was an opportunistic experiment because we happened to have some suspected amylidinium-related mortalities. So we decided to see how that would affect the overall microbiome following copper treatment. So we focused on metal resistance genes and antibiotic resistance genes as well, and the differential expression of those. This is an overview of those results. The first graph being showing some, we focused on what we consider opportunistic pathogenic genera, which include Vibrio and Pseudomonas and a few others, just to kind of narrow down the view. However, there was no significant difference. in the presence of these guys, except in one of the treatments. Sorry. So, and then the graph in the middle shows you the metal resistance genes, which, I'm sorry. I'm sorry, I remember the main takeaway that we wanted to get or want you to get from these graphs is that I forgot to mention we sampled biofilm. So the biofilm could be a reservoir for metal resistance and antibiotic genes, which can be co-selected together in opportunistic pathogens. So that was an interesting observation. The next one that I'll go over, we did a while ago, was our the first on-farm research project that I got to be involved in. We focused on larviculture at different salinities. We had 3 different salinities ranging from 10 to 30, and we took a whole bunch of different kinds of samples throughout the experiment period. And here is just a quick overview of those results because this project has been presented previously. So this graph is an overview of the microbiome in the tank water. And we did see significant influence based on salinity in the microbiome. So we expected that as well, since some bacteria are tolerant to lower salinities or higher salinities. And then here is just some gene expression graphs. We focused on some genes that are associated with oxidative stress. However, only 1 of 6 genes was significantly different between salinities. And we did see a rise in specifically a catalase gene. So a lot to go through, so I'm sorry. I need to go a little quicker and skip over some details, but so that project focused on larval, we wanted to do kind of a part 2 focusing on juveniles. So in that, this was done at Harbor Branch, and we focused more on the presence of Vibrio species, and then the immune related genes in organs. So we were like trying to focus more on different areas. So we took gill, liver, and spleen samples. Throughout the experiment, we had positive growth for different types of Vibrio. We used a culture medium that differentiates and selects between these species. So we also developed a qPCR assay for specifically Vibrio and Gualarum in this process. Quick results that are hopefully going to be published soon. This is just a graph for some differential gene expression. So these 2 genes, again, are associated with either osmoregulation or immunity, but there wasn't any significant difference between the salinities. So there are also no signs of vibriosis, which is good. And basically, I mean, this negative result is good because that just confirms that Flora pompano is really tolerant to changes in salinity. Okay, so now we're moving on finally to objective 2, and this is Finfish genetics for selective breeding. I'm sorry, I have to take sip water. Okay, so just to refresh our memories on the selective breeding process and how genetics can help facilitate that. Our main goals are to provide, first, to provide insight on the genetic diversity of wild populations of pompano and red drum. So I will be talking about both species for all of this. So we sampled, well, actually, I'm sorry, that's the next slide. So we want to first look at the wild population genetic diversity. Then we plan to improve the genetic quality of fish stocks. I'm sorry, for superior performance and production. And I'm going to try to stop saying, but okay, so this graph, I'll point out first, is just kind of going over how we would calculate the environmental gain and the genetic gain over one generation. And from this, we can gather that with only the environmental factors, the gain would be here. With genetic improvements included, the gain is here. So this is based on size. And so that shows us that it's a quicker process. We can zoom in on economically desirable traits, being faster growth rates, larger size. We can also attempt to control sexual maturity and improve the efficiency of production. So we are hopefully, what we're working on doing this with DNA marker assisted selection, MAS for short. And so this would help optimize the selective breeding program in a variety of different aquaculture systems. So it can essentially be applied to species other than these two and systems other than RAS. So here's just an overview of the genetic diversity we've seen so far in a subset of wild populations. So Florida pompano, for both species, we sampled various sites with our collaborators. Lots of people helped. So when I say we, it's like a big we. We sampled areas along the east coast of Florida and the west coast of Florida. So lots of diverse areas in general. So we would expect to see a higher or greater difference in genetic diversity between populations that we sampled on the east versus populations on the west. But we did not see, based on our preliminary SNP analysis, we did not see significant differences from east to west coast for either species. We're still in the process of adding more samples to the data set, and this will help us in really confirm that. So this is just some pictures of us sampling at Aquaco. Joe in the back there, and Aaron somewhere, but he's always around. And then here is just a couple pictures of us extracting DNA from the fin clips that we take from the fish. And this is a poster that was just presented at UF by one of our grad students. She focused on different head sizes in Florida pompano. So that would be a more specific part of our growth marker experiments. And I'm sure I took way too long. So if you have any other questions, or if you have questions or want to know anything else, let me know.
I'll go very fast on this, but I don't want to lose the, I want to go without saying that all this work is a skill, several publications, student awards, and we have workshops on Florida Pompano. And last year we were granted a full session in the WAS in San Antonio. So I'm proud to say that. And here are some of the publications that we have, and you can all look for them regarding Pompano, influence of product nutrition, or the status of Florida Pompano, methodological changes in Florida Pompano. Also, we have recently published in this Aquafeed magazine for black butterfly meal, and some awards with our students in the IRSC. And here are some of the workshops online, and here are all our YouTube presentations during the WAS last year. And lastly, I want to have a future research and our projects that we're working on at the moments are a challenge facility, a selective breeding facility, a brood stock maturation facility, a rapid in situ nutrient detection, and a feed production lab. So rapidly, this will be our future disease challenge facility overall, with a concern to assist stakeholders to identify and treat diseases too. Here are some of our selected breeding facility, which is are still under construction. And here our stock maturation facility also under construction. And one thing that we are working at the moment is we're also working on developing a rapid in situ fatty acid detection to determine egg, larvae, and fillet quality correlated to the fatty acid content through the ramen. Lastly, we are building an aquafeed production facility, and we acquired this new extruder so to produce high quality pellets, and also with the perspective of becoming a call center to formulate and provide universities, research center, or even stakeholders with any kind of feed size or texture. So that would be all for our presentation, and thank you everybody for paying attention to this.